Welcome back to yet another Journal Club brought to you by Lifespan.io. I'm Dr. Oliver Medvedik, and today we have a short paper to discuss. Not a primary paper, it's a review paper from a lab where I did my PhD way back when, Dr. David Sinclair. And let me share the screen without much further ado. So this paper is about a theory of aging. There's lots of theories of aging out there. Eventually, we'll have one grand unified theory of aging that, encompasses them all. But right now, here's one perspective. I'm going to share my screen here. It is the information theory of aging, or abbreviated ITOA. Is it pronounced ITOA? I'm not sure. But it is the information theory of aging, brought to you by Liu, Tian, and Sinclair, published sometime last year. And basically, there's a lot of talk about what's the key driver of aging. We covered, a review paper way back when on the hallmarks of aging, all the different ways that things can go wrong. And fundamentally, I think there's a dichotomy of opinion of what aging is, or what's the key driver of aging. And I think if we kind of go all the way up in the tree of hierarchy there, some people think it's programmed, some people think it's not programmed. This basically falls into, I think, squarely the not programmed camp, which is a loss of information is the key driver of aging, right? And I fundamentally agree with that aging is an increase of entropy in a system, and the system has ways of trying to counteract this accumulation of entropy. The focus here, though, is what's the key kind of source of entropy. And here it's David and company is speculating that it's essentially this loss of information is fundamentally at the epigenetic level. So we've covered a lot of papers here where we talked about cellular reprogramming. So this review paper here has a number of schematics, and what David here is contrasting is the loss of epigenetic information with other types of entropy or losses of information that happen. One other hypothesis as to one of the key drivers of aging is a loss of genetic information, which is basically point mutations and other types of mutations that happen in your genome, leading to all kinds of things that can go awry, notably cancers, but also progerias and so on and so forth. And then utilizing something like gene editing to repair that. We do see an increase of somatic mutations over time in cells, and we have covered a paper in the past where somatic mutation rates correlate inversely with the longevity of an organism. So there's something to be said for that. What's interesting, though, is that number one, epigenetic changes happen as well. So those of you who've been following us know that there's lots of ways to measure this. One of them being a variety of clocks. One of them eponymously named after Steve Horvath, which is the Horvath clock, which looks at methylation status and its changes throughout the genome. And these can be very highly predictive of the biological age of an organism. So these changes, or epimutations, as are shown in this figure here, accumulate over time. But unlike losses of genetic information, which is sort of hard-coded, and in David's parlance here, this is sort of digital changes versus the analog changes in the epigenome. And I think David here casts a wide net. He refers to I believe the epigenetic changes, not just methylation changes, but obviously there's many other markings that can happen on histones and the DNA as well, not just methylation and acetylation, but there's I think at least they've been cataloged over 100 different types of chemical changes or chemical groups that are added, and it's still kind of early days to figure out how these are affected by aging. But there's also other architectural changes that happen within the chromatin, where the DNA basically is aberrantly remodeled, right? So the key here is that you can reverse these epimutations, these changes, by subjecting the cells to um, reprogramming factors. So these Yamanaka factors, so OSK, and you can also emit c-Myc. If the cells are essentially treated with these factors for a period of time, you can essentially revert or reset these cells and get a rejuvenated cell. So it's very tantalizing to speculate that this may be the main, or maybe maybe not, perhaps the only or the key driver of aging, because what people have noted is that you can get, certainly in petri dishes, you can get full rejuvenation. If you clone animals, you can do this over and over again. So you basically can essentially seemingly reset the clock over and over again. So is this really the key driver? Another figure here basically illustrates, one way of looking at it, these epigenetic changes, is that you can have two ways to revert them. One is partial reprogramming and full reprogramming. So when the Yamanaka factors were discovered in around 2006, 2007 by Shinya Yamanaka and partners, they were looking at ways to reset essentially cells from a fully differentiated state, which is shown here in this figure as defined identity. So this is basically this, this is a classic figure of a differentiate Waddington landscape, right? Which was, I think, posited a long time ago. When did Waddington come up with this? I think it was like in the 50s or 60s. It was a long time ago. So basically, cells go from a very non-differentiated state to a differentiated state. So, stem cells of various stemness finally end up in a terminal state. But this state doesn't stay in this position. As aging occurs, further changes can happen, which is referred to here as X differentiation. So you have a loss of identity in this direction, where the cells are fully, in a stem-like configuration and can become any type of cell, to a different type of loss of identity, where essentially the reading of their genome becomes fuzzier and fuzzier. So essentially a loss of information. And then the question is, what is the key driver here of this loss of information? And it's posited here that it's epigenetic changes that are the key driver. Supporting that is this concept, or this treatment actually, which has now been done in mice, of not only full reprogramming, where you essentially use these Yamanaka factors to get a cell to go all the way back to a undifferentiated state from a differentiated state, but you can take cells that have lost their identity as a result of this X differentiation, aging, and also drive them to a fully programmed state, or you can carefully dose these factors and get them to a partially reprogrammed state, which is back to their defined identity in a fully differentiated state, but in a youthful state. So in both of these cases, in full reprogramming and partial reprogramming, if you then take a look at these cells and examine them through a number of different parameters using Horvath clocks, various physiologic metrics, mitochondrial function, it seems by any parameter that you look at, these cells, they're in a much more youthful state, or essentially reset their biological age, just through essentially the introduction of these Yamanaka factors. So the question is this now really, is this loss of information primarily epigenetic, and is it all fully reversible as by using these reprogramming factors, either through full reprogramming or, as is now being done, through partial reprogramming? And you can get substantial increases in mouse lifespan, in a variety of experiments that have been published so far using these techniques. So I'm going to stop sharing right now. Another interesting aspect to what David mentions in this paper is DNA damage and how it can drive epigenetic reprogramming. So it's noted that if you have double-stranded breaks, you have other types of damage. What can happen is you can have a variety of repair factors migrate from areas of the chromatin and go and try to repair that damage. Sort of as a secondary consequence that may be a key driver of this is that that reshuffling of factors might leave other areas of the chromatin prone to epigenetic, I would say, mismodeling, and that would be a further driver of epigenetic changes and X differentiation. So in a sense, it's not the DNA damage specifically that is causing mutations to accumulate and is driving aging, but the response to repair the damage, which can drive other areas to lose information via the epigenome. Now, one kind of important corollary to this that David mentions in this paper here is if you can reprogram the cells back into a youthful state through the introduction of these Yamanaka factors and other variations of them, that sort of begs the question: where are these factors getting the information to perform the repair in the first place? So if entropy is increased in the epigenome throughout the cell, I'm going to share the screen here, and you lost this information. So this is basically just a summary of the types of information lost that are recovered by epigenetic reprogramming, right? So here's a nice figure that you could look up all the, so, histone levels decrease as aging occurs, and then these are the interventions, so a variety of different, either LSK or combinations of OSK with other factors, which are listed down here, OCT4, SOX2, KLF4, MYC, Lin28, NANOG, right? So methylation changes happen. There's all sorts of other types of changes, but these are all reversed in a variety of different cells, both mouse and human, in vivo and in vitro. If this information is being recapitulated, then there has to be a way for the cell, even though it's lost that information supposedly, there has to be a way for the cell to know how to get back to the original state. So David here speculates, just scrolling through here, these are just other variations on this theme, speculates that there must be a repository of youthful epigenetic information somehow encoded in the cell, in the nucleus, obviously, where these factors could essentially use that information to recapitulate, the original state of the cell. And I think that's an interesting way to look at it. That's the first time that I've heard that mentioned, although that may have been mentioned before. So if you are reprogramming these cells and the cells are X differentiated, they're aged, and the information is, sort of gone, how do these factors know how to reset the system essentially back to the state that it was, right? I've got my own thoughts on this, and I think maybe I can open the mic to anybody else who wants to comment.
I can't. That's it. Well, it's all very interesting. But you know me, Oliver. I'm very excited about the whole idea of partial cellular reprogramming.
Mm-hmm.
How does it fit? How does this fit? Well, like I was waxing poetic before we went live. I think it complements the established sort of hallmarks of aging quite well. I will say that I think we've got to establish whether it's how primary a role epigenetics plays, in the context of aging. It's like the 9 hallmarks, or whatever system you prefer, the SEN system,
Mm-hmm.
whichever. I think it's still yet to be sort of proven definitively whether there's actually any primacy. I know hallmarks as it primary, secondary, and tertiary hallmarks, but I don't know. I don't want to be too optimistic, but yeah, I'm quite bullish about cellular reprogramming. I think it is the biggest bang for the buck. So yeah, for me, I quite like this theory. it makes sense. Because a lot of people used to think that it was like mutational load that was more important than epigenetics, but everything I'm seeing, I think epigenetics or gene expression plays a significantly more important role. But I might be wrong.
Yeah. So one thing, I think types of experiments that would, really elucidate this are ones that are a bit more quantitative in nature. So if it's the loss of information, we should be able to quantify that, right? And so what I mean by that is what's the contribution of the epigenome and changes of the epigenome towards, the aging process in general. So if it's the key driver, if it's the main driver, then you should, in theory, be able to keep resetting the epigenetic clock in a mouse and have the mouse live forever, right? So just basically keep it going eternally. I have a hunch that's probably not going to be the case, but unless we set up the experiments to do so, and one type of experiment we're actually collaborating, we're having some discussions with Vadim Gladyshev to try to set up some experiments to answer this, which is, could we either in vitro or eventually in vivo have a mouse system where you essentially have OSK factors pulsed at a periodic rate throughout all cells, and what's the contribution of these epigenetic changes? And another key question is can these effects can partial reprogramming maintain its efficiency with each subsequent partial reprogramming session, right? So we know you can partially reprogram to an extent, but can you do that once, twice, 100 times? And is there, diminishing returns after a period of time? And if there are, that would suggest that there's probably other drivers out there as well. So, those are experiments that still need to be set up, I think, to quantify what's going on. The other question I have is when people do epigenetic reprogramming, either partial or otherwise, I haven't done it myself, but from what I read is it's fairly inefficient. So it's not like you reprogram every single cell in a petri dish. It's a subpopulation that manages to successfully reprogram. So then my question there is that David posited in this paper that there must be a repository of youthful information, because otherwise, how do the reprogramming factors know? What do they use as a template to go back to the original state? And that might not be as much of an issue if perhaps most of the information is still somewhat intact, and could basically be filled in. So in other words, if you have changes in the epigenetic landscape, I'm just gonna throw out something here. You have two methylation marks normally at a location, and after a period of time it drops to one methylation mark. Well, the cell could look at that one methylation mark and say, well, obviously there needs to be a second methylation mark there. So even though you've lost 50% of the information, and that's would be a massive change if we extrapolate that to a whole bunch of other locations, there's still enough information there present to reset it back into its native or youthful state. But if you lose too much of that information, if now there's no methylation marks at that location, you can't partially or fully reprogram that cell. So that cell basically dies, right? So it's only the ones that you have, only the subpopulations that have a sufficient amount of information still intact that could be fully reprogrammed. Again, that's kind of my hand-wavy thought process here. So unless we do experiments to kind of quantify that and figure out which cells are actually being successfully reprogrammed in, let's say a dish or in vitro, and why those cells are being reprogrammed versus other cells, I think that might tell us some, no pun intended, interesting information as to what information is needed to remain to get to reprogramming to successfully happen. So I think that's one issue that wasn't raised in this paper, is how in practice reprogramming happens when you actually do it. And it's, to my understanding, it's an inefficient process in that there's a bottleneck. You only get a certain subpopulation of cells that actually make it through. And then I think a key question is there, what is special about those cells that allows them to be reprogrammed and not the majority of other cells? Yeah, DDA.
Yes, first, thank you. This is quite interesting. I have a few comments. Maybe my first always depressing comment is, as far as I know, until now, I know that David Sinclair is always saying, okay, we can reverse aging of mice and so on and so on. But as far as I know, there was not one experiment where you are applying this method and that mice are living longer. That's the first point. The second point is even, let's say, even if this theory is correct, there is still the fact that animals age at different rhythms. And so it means that this epigenetic age is programmed, because for some animals it will be happening in, I don't know, 2 years, and for other animals it will be happening in 100 years. So, okay. And then I had a third comment, but I forgot. Okay, I will be back when I think again. But yeah, can you confirm or not about the fact that there are no experiments until now, and maybe also concerning what you were saying, that so if I understand correctly, what you say, Oliver, actually you cannot. Let's say, have the Yamanaka factors working in all cells of the mice, and not for humans neither. But just to know, to understand.
Well, I mean, yeah, there's certainly technical difficulties there. I don't know if there's any biological reason why you can't have Yamanaka factors working in all cell types, but certainly, with using viral vectors and other ways of delivery, you're not going to really get 100% penetrance to all cell types, depending on your delivery method. I think that's still an issue, right? So you certainly can, using these viral vectors, infect, liver cells pretty readily and other cell types, but not neurons, for example, or muscle to some extent.
Yeah, and I interrupt you to say neurons are, of course, very important. I mean, if it's not working for neurons, it's a bad sign. Yeah.
Yeah, I mean, it's still very positive that you get lifespan, despite the imperfection of getting these factors delivered to all the cell types and all the tissues. Despite that, the fact that you get a significant increase in lifespan and a reversion to youthful characteristics is extremely promising. So that certainly suggests that you would see even more dramatic effects were you to, get this to happen in every single cell type. Now, the question is this, can this be maintained in perpetuity? If this is not only the main driver, but the only driver of aging, the loss of information, primarily through the loss of information in the epigenome, then you should, in theory, be able to, have the system going on forever and have essentially an eternal mouse. Or if that's not the case, then over a period of time, you will still, the animal will still age, or there'll be some aspects of aging still accumulating that are not epigenome related. There'll be still there'll be still an accumulation of entropy or a loss of information, loss of biological information over time. So I think that's really, that's an important question for this theory here. And experiments need to be designed to basically help answer that.
I interrupt again to say, well, as far as I know, there is not even one experiment just prolonging the life of mice. It's only in petri dish or
No,
never.
they've
Yeah.
delivered these. They have delivered these factors in vivo in mice. Yeah.
Yeah, and it was what? How many percent more? I forgot. Then.
Well, according to Dave, well, so I think they were delivered late in the age of the mouse, so it wasn't started very early. It was delivered.
Yes, no, I
Yeah.
remember. Yeah,
Mhm.
yeah. No, I remember about this article. Yes,
So
yes, yes.
so
I
when you
remember.
so when you say you got like 100% extension in life, it's I think it's 100% after a
Of
certain
a very
of a
short one. Yeah.
of a
Okay.
after a after the mouse was like late middle age or later in life. So if you didn't do this intervention, they died at this point. If you did this intervention, they died, it lived twice as long at that section that they monitored. So they have done in vivo experiments to an extent using viral delivery vectors.
Yeah,
Yeah.
I forgot about this, and this one is indeed very promising.
Mhm.
I know that Aubrey de Grey was saying, like you say, yeah, but it was already very old mice.
Mhm.
But yeah, still it is indeed quite promising.
Yeah,
And
it's
it
actually
was three of the four factor Yamanaka factors.
yeah,
Yeah,
and
okay.
so in a way, it's actually even more promising because the fact that you can get that extension after doing the intervention late in the life of the mouse is quite promising. Meaning that again, that the information is being reverted to a youthful state. So even though the mouse is old, the cells are old, there's still enough quote unquote information content somewhere, scattered throughout the epigenome that could be used as a template by the factors to reset it back to its original epigenetic pattern. So again, how that information is still there, in this paper, David posits that there's a youthful repository of information that that could very well be the case that that you have regions of the epigenome that are intact and don't change and are in a sort of a protected or sequestered heterochromatic environment. That's one option. Or it could be that that you don't have full loss of information in a subset of cells, because not every cell, I believe, gets reprogrammed. Some of them will just die. But the ones that do get reprogrammed, there's enough extant information there that just as if you have bricks missing in a building, you can tell with the surrounding bricks that a brick needs to go there, right? So even though you can have 50% of the bricks missing, but looking at the pattern of the rest of the bricks, you can see, well, I know where the bricks are supposed to be, so I can just put them right back into that place, right? So but obviously, if all the bricks are gone, you really don't know what the pattern is anymore. So to some extent, if you have too much information loss, you can't use the rest of the remaining pattern to fill it in. So I think we kind of need to tease apart exactly, how the Yamanaka factors are using the information that's present to essentially reestablish the information that was lost. So I think a lot more careful experiments need to be set up to quantify this. So that's my take on this fundamentally.
Oliver.
Yeah.
Just to circle back on what you've just said, are you suggesting some kind of like information or shared information network that fills in these bricks, almost like the cells are a Gestalt entity?
Like the cells were a what entity?
A Gestalt entity, which means
Gestalt, like
a Gestalt is a German word to describe, I think, typically like a monster that's comprised of various different personalities or individuals, a Gestalt being. But in this case, it's like a collective group of cells or tissues contributing to an overall picture of that information and restoring it that way. Is that what you're suggesting?
Not really. I think I'm suggesting it more on a let's I'm going to start making up terms here. A Gestalt unit. So if we can look at all the changes that are happening, all the information loss, I think it might be interesting to see if we look at one specific area the chromatin that gets remodeled by Yamanaka factors and all of the other things. So the Yamanaka factors switch on other factors, which then do the remodeling, right? So you've got this cascade of things happening, and areas basically get reprogrammed. So if we can look at like one tiny area where you have X differentiation, as stated in this paper here, a loss of information. I'd be interested to see if, how much information is still extant in a certain area. And if there's too much information loss in a certain defined area, can reprogramming happen in that area? And if not, then what is the minimal amount of information that is still needed? By information, I'm referring to methylation status and probably other types of epigenetic modifications, both on DNA as well as histones as well. But how much information is required to reestablish essentially, the youthful state of the differentiated state of the cell, and also how exactly do those factors that reestablish those epigenetic modifications, how do they use that remaining information to reestablish the baseline that was lost? So I think that's, and so there must be some kind of minimal, process there, or a sub-process. So there's a minimal size to that gestaltness, I guess you could say, right? And then you can expand that to holistically to the rest of all the chromosomes and all the epigenetic changes. But we can, certainly, I think we could design an experiment where maybe we look at various tiny areas and see, what's the minimal amount of information that needs to remain. Is that information still encoded in that area that gets changed, or are there perhaps other templates, of, as David mentioned, youthful repositories of information somewhere else that interact with either the Yamanaka factors or downstream, anything downstream that these factors then will express and cause, chromatin changes to be, reestablished back to the way they were. I haven't, I think I'm going to do some digging and see if these types of experiments have been done. I don't think so. So yeah. So now I'm intrigued to see if people have looked at specific regions and see how exactly the system is reestablishing or resetting itself back to an earlier state.
So we have the birth of the Gestalt networks, which the description of that word is a structure, arrangement, or pattern of physical, biological, or psychological phenomena so integrated as to constitute a functional unit with properties not derivable by the summation of its parts. In other words, it is a combination of things that is greater than the sum of its parts. Interesting. We know that the body has ways of preserving information. Look at the germline cells, for example. They're very highly protected. They do get damaged, but they are protected in order to conserve enough information, as you were alluding to, for the next generation. So they do get damaged. There's no doubt about it. So yeah, there's got to be some kind of recovery or backup system. I would say that a network like that, a Gestalt network, that's such a buzzword, isn't it? I would say that's quite sensible in a view of actually preserving information, if that makes sense. Mm-hmm. So if you take out one area of the machine, it can still work. There's lots of redundancy and backups built into it. That would seem to be quite logical as the way to design a robust life form. And I think we're fairly robust, right?
Mm-hmm.
So
yeah. So again, whether or not this is the main or the only driver of aging that's yet to be determined, and like I mentioned earlier, we should set up experiments, and those experiments can be set up to answer those questions. My hunch is, I'll go on a limb and say that it's not. I think examples where you have organisms, for example, hydras and other organisms of that nature, where The cells are very plastic. A lot of reprogramming takes place, and these organisms, they asexually reproduce by budding. They're quote unquote immortal. I've mentioned this kind of before, like when I think about this in the context of what's immortal. Yeah, that organism might be immortal in toto, but those cells are very mitotically active, and I bet a lot of damage is being exported by cells basically that are kind of too far gone with their information loss, essentially dying off or being shed, right, by the organism in such a way. The resetting of kind of an entropic state to a lower state of entropy kind of, well, implies that entropy can be removed. Either it could be reset or the entropy is basically kind of, jettisoned out. There are types of entropic accumulation, I think, in higher organisms such as ourselves that might not be so readily removed, such as things that happen to our extracellular matrix, right? Cross-linking, for example, protein aggregates that accumulate. I can see the resetting of entropy when it comes to information in the epigenome being, you can keep, as long as the information loss isn't too far, you can in theory keep resetting it forever and ever and ever again. But there might be other forms of entropy that accumulate, such as the ones that I mentioned, that our metazoan system doesn't have an outlet to remove, right? We just don't have that, and that might be another engineering challenge, right, to overcome. So as you reset the epigenetic status, the cells are basically still kind of stewing in a sea of entropy of other types of entropy that's accumulating around them, and that will then serve to dampen the resetting process, right? Because the cells are basically sitting in their own muck, and we don't have, that system doesn't have a way to remove it. Now, in an organism such as a hydra, that might not be a problem. The cells divide and kind of slough off, and the entropy gets jettisoned out into the sea of where they live. But ourselves, we mammalian organisms might not have that capability. So I think fundamentally, aging to me is not programmed. So when we say programmed and not programmed, yes, there are maximal and average lifespans for different species, but that's not really what we mean by programmed. There's not a genetic circuit that that is evolved to essentially switch on the aging process. Rather, we have systems that can reset the system and repair processes that promote longevity, but they in themselves are essentially victims of entropy as well, right? So, and that basically drives the whole process to a further and further entropic state. So basically, yeah, what I'm saying is I'm going to go out on a limb and say that because of that, I think epigenetic changes are very important. I think we'll see that there's a lot of resetting that can happen to an earlier state. I think we need to find out how that information that's still available for reprogramming to successfully happen, how it's maintained in the cell. What's the point of no return? How much information loss can be tolerated before there is not enough information to be reset, whether or not this information is in certain areas that are repositories of youthful information or distributed, as I mentioned. I think that needs to be quantified. But I think at the end of the day, depending on the organism, right, full rejuvenation might happen in a hydra just through epigenetic reprogramming, but in other organisms, there might be other layers of entropy that need to be dealt with. Full reprogramming will get us. I'm just going to throw out some numbers here that have no scientific backing, but full reprogramming might get us to live to age 150 or 200, but then other things will kind of screw that up because there'll be accumulation of other entropic events, and then at that point we can start to quantify and say, well, what are those other forms of information loss or entropy gain. And you can make a pie chart and say, well, for humans, I'll just make up some numbers here. It's up to this point, it's 75%, these epigenetic changes, loss of information, but there's these other aspects of information loss that are happening that will then dampen any positive effects that you get from from resetting the epigenome. So again, these are still unanswered questions, but I'm actually excited that we're even able to have this discussion here and think about quantitative experiments in that direction. It kind of tells me that this field has come a long way. I certainly think of it, and I keep thinking about it as David has done in this paper, that it is, it's primarily a loss of information. We could just say that, generically it's a gain of entropy, but there's many different types of flavors, if you will, chemical flavors of entropy. And I think the epigenome changes are one of them, maybe a major one of them, but I think not the only one. So, yeah. Didier, hands up.
Yeah, just actually the same quick comment I did for me already. Okay, I agree with more or less everything that you said, except one. For me, it's clear that the rhythm of epigenetic change is programmed, because you see difference between animals, and because you see animals who seem to have no epigenetic change, or who can get rid of this. And this is, of course, programmed, but actually it's not so important to know if it is programmed or not. The important thing is to try to change it. That's it.
Okay. I guess again, coming back to the terminology of programmed versus non-programmed, maybe I have to sit down and write out exactly kind of what I mean when I refer to programmed and unprogrammed. So I guess fundamentally, evolutionarily, when somebody says that aging is programmed, then that implies to me that at some evolutionary time point, nothing aged. You had cells, essentially, the first primordial cells that developed, aging just didn't exist. The cells never dealt with the loss of entropy. There was never any problem. And then evolution somehow started putting in mechanisms to drive entropy forward in cells. And I think that's, I don't agree with that assessment. John?
I agree with what you said about Hydra. I actually studied Hydra in grad school.
Mm-hmm.
And they do have this constant cell division, and they slough off the outer layers.
Mm-hmm.
I also agree with what you said about extracellular matrix. It just is a chemical reaction. The glycation cross-linking occurs very slowly over the course of aging. I'm not quite sure about David Sinclair's idea, or maybe the analogy is just difficult to follow. For example, entropy and scratching of CDs and things like that does not convey to me a lot of biological understanding. And I think it's just such a vague set of terms that I'm not sure that it's useful for us to use those terms. Maybe we need to find other ways of describing what's going on.
Although I will chip in and say, John, that I think David presents it like that mostly as a public-facing thing, so that people, average people can get on board with the concept. I mean, of course, cells are not CDs, but here's the frightening thought, though, and it's a sign that we're all getting older. A lot of people go, "What? You used to listen to music on those things?" So it's like, what about cassettes? But I mean, But joking aside, I think that's the analogy. It's like people call senescent cells in the popular press, they call them zombie cells. It's not really accurate because we know that there's a lot of nuance now to senescent cells. Some of them apparently are quite useful. Some of them are required for things. So just nuking the entire site from orbit ain't a great idea. But yeah, I think it's interesting. And I was just going to say that I agree with what Didier said about, yes, some epigenetic changes can be random, for instance, epigenetic drift, but also some of it could be more programmatic in its nature, but it doesn't ultimately really matter as long as we can reverse it. So I happen to agree with what Didier said there. It doesn't, I mean, it's interesting, but let's develop the tools and actually be able to just reverse it, and then it doesn't really matter where it's coming from, right?
Yeah.
Well, when I think about programming, I think about puberty, for example. We start out as little kids, we start growing, we go through puberty, a lot of changes happen in our body, and we stop getting taller, for example. And I wonder if the thymus degradation at age 60 is a kind of programming, and if so, how would we maintain our immune systems? And I think Greg Fahey is working on using testosterone and a combination of other things to maybe reprogram thymus growth. I wonder what you think about that.
I was going to mention earlier when Oliver was speaking about the programmatic elements and things. I think it's worth a shout out to say that there's also another theory called the developmental theory of aging. I think it was João Pedro de Mengues. Sorry about butchering your name, Pedro. I'm trying. I think it was Pedro's theory, the developmental theory, and that is an interesting way of presenting the programmed side of aging. I'll give you an example, Oliver. I know you've got that I'm not convinced face, but I'm going to talk about salmon. Okay, salmon.
I was just thinking about that. Yeah.
Yeah. So salmon kind of have that programmatic element in their life cycle, and as John was saying about like the puberty, adolescence, there are different distinct stages in human development. Obviously sexual maturity being the nature's goal. So why not? I mean, I honestly think that it's kind of both, but the program element is something that people don't necessarily agree on. So even the programmers don't agree necessarily what program is, but I definitely think there's programmatic elements in epigenetic changes. Of course, there's things like environmental aspects and things like that, and damage, such as random things occurring like epigenetic drift as well. I mean, that's a thing. Telomere shortening definitely contributes to changes in epigenetics. So nothing works in isolation here. So I honestly think that we've got some really interesting, more modern theories, and I think that everybody's a little bit right about something. And importantly, as you and I have talked about before in the past, I think we are moving sort of closer towards that consensus. It's like, it's not fair, right, guys? Because all the other big science branches have got these big general theories like physics. And, we do not have a general theory of aging yet. I would love to see that happen, but I think we're getting closer to it. And things like this and the developmental theory and the deleteriome, they're all sort of building on what we already know. And I think eventually it's going to sort of, that's how it's all interconnected. So I think everything's a little bit right. I know that's really vague, but I do.
Well, I think fundamentally, I mention this to my class all the time, that, biology has some big holes to fill, and fundamentally one of the biggest holes isn't necessarily aging, but it's origins of life in general, right? So that's you need to put life on a sound chemical and thermodynamic platform as to how it originated and what were the drivers, and we don't have that yet, right? So it's still a bit shaky. We're getting there. I think I have full confidence that there will be physical chemical laws that we will establish where we know what's happening, and that because it's kind of hard to accept the alternative, which is a miraculous event, right? So that's the only other alternative, and it's sort of, scientists don't like to go in that direction. You kind of at that point you've thrown your hands up and given up, right? So I don't think that's an option for
I hope we're not going to
working
drift
scientists.
into intelligent design.
Well, that's the only other viable option, right? So we're going to have to ignore that for the moment. So,
Yes.
but coming back to what you were saying about programmed, to me fundamentally, I guess I'll just kind of, for me, I won't talk any further about that concept, but for me, concept of programmed aging is really all of those are fundamentally different versions of antagonistic pleiotropy, which is the concept that what helps you essentially procreate early. And so what gives you the most offspring is what's going to drive evolution and what's going to essentially promote the existence of your species. That's it. That's all evolution. So if you do something stupid and it kills you after you procreate, fine, fantastic job, mission accomplished, right? So if the salmon dumps a ton of cortisol into its bloodstream so it can swim upstream and basically turns into just a pile of accelerated aging mush, just so it can spawn, great, fantastic, it's done its job, and the system, the population, the species, right? So the population of cells within the salmon, the somatic cells, are kaput, but the population of salmon as a species keeps going on, on and on. So in that respect, yeah, aging can be programmed, but I don't really see that as really programmed. I see that more as different facets of antagonistic pleiotropy to essentially keep the species from going extinct, right?
Well,
So
can't antagonistic pleiotropy be programmed? But I want to
yeah, sure. Yeah,
or
it
programmatic,
yeah.
then, should we say? Should
Yeah,
we say programmatic, because that's what Pedro says.
Yeah.
He doesn't say programmed aging. He
Yeah.
suggests that there are programmatic elements.
So when I hear programmed, so I read a book some years ago that was essentially, it was published by an immunologist. It came out in the 1990s, and I vehemently disagreed with what the author was saying, who is a respected immunologist. Basically, almost talking about aging being programmed, but in a more fundamental sense, which is that, like I said earlier, that single cells like bacteria, which we know do age, they do accumulate damage, but that wasn't known at the time, don't age, and aging evolved later on in the evolutionary of life. So in other words, entropy for an individual cell just doesn't exist, and evolutionary mechanisms had to come in and say, oh wait, this is too much of a good thing, we're going to have to put in systems to let you guys age.
Really? This was in a paper? I mean,
Well,
I'm not buying it.
No, but fundamentally, when you get to the heart of it, when you talk about programmed aging, right? To me, that means evolution had to come up with the concept of aging, and I disagree with that. So that's what I hear when I hear programmed, not programmatic when it comes to antagonistic pleiotropy. Yeah, those elements, sure, but to me, that's not programmed aging. That's programmatic aspects of antagonistic pleiotropy that can basically accelerate the demise of somatic tissues because you need to procreate and maintain the species. But what I'm saying is that the loss of information as a result of entropy was always present. It didn't have to evolve later. This is something that cells have to deal with on a continual basis because it's a physicochemical system that's bound by the laws of thermodynamics and entropy accumulation.
I understand thermodynamic entropy, but I'm not sure what you mean in the context of aging humans.
In the context of aging humans?
Yeah. In other words, how does the concept of thermodynamic entropy cause any aspect of aging, and what is entropic aging and what is non-entropic aging?
I
For
would say,
example,
well,
is cross-linking of extracellular matrix, is that entropy?
yes,
Or
yeah,
is that okay?
yeah, I would say all, so if we have a system that's at perfect homeostasis where it can do all of its everything, it's theoretical optimal parameters, whatever they are. Your collagen is at its optimal elasticity and tensile strength. Your everything is, to me that is at the entropically lowest state. So when I say the information state, I'm saying the information state of the tertiary structure of the proteins, the information state of the epigenome, so on and
Yep.
so forth. And anything that deviates from that, where the homeostasis becomes less efficient, is a gain in entropy. And that could be either reversible if it doesn't get too far, or at some point there's too much of a loss of information for it to be reversible. So when I say entropy and loss of information, that's what I'm referring to. Loss of that chemical information that allows homeostasis to be at its optimal.
Yeah. So as I put in the chat, it's basically just like the universal rule that planets and everything obeys. It's the movement of order to chaos, and it happens on a biological level too. I'm going to say that this is a pet peeve. I don't hear it as often now, but a lot. About 10 years ago, people used to say, oh, you can't reverse aging because the second law of thermodynamics. It used to drive me crazy because it's like, tell me you don't understand the second law without telling me. Because obviously the good news is that is an absolute true fact. Yes, but only in a system that is closed. But biology is not a closed system. So therefore it's subject to intervention. So good news, everybody at home who's watching. There's no physical law or reason why age reversal is not possible. So if things can go from order to chaos, they can go back from chaos to order. There may be a tipping point with human aging. There may be a point of no return, like Oliver's just mentioned. But in general, it's probably pretty far down the line before enough information, see how I segued that, is lost so that it can't be recovered. So there you go. But I used to hate that when people say thermodynamics.
I mean
Drives me mad.
I mean the perfect rebuttal to that is if that was true, then we couldn't have babies.
This is exactly true, and this is what
it happens.
People say,
all the
"Oh,
time.
it's not possible," and I'm like, I go, "Really? Oh, okay then. Have you got children?" And they say, "Oh, yeah." And I say, "Well, were your children born old?" And they were like, "Well, no, of course not. They were born babies." So I said, "There you go. You've just ran your own personal age reversal experiment." And thousands of them are made every day. It demonstrates that information can be preserved, yes, via the germline. Yes, it is very well protected, but it does get damaged. We know it gets damaged, but enough of that information survives to actually create a whole new person. And it only takes those 4 programming factors that we're talking about here.
Yeah, and
amazing.
And related to too far information loss and kind of, how, resetting the aging clock, in babies, again, this, nobody's done these experiments to correlate this, but I kind of view it this way when it comes to reprogramming somatic cells in a dish. Some cells can be too far gone. I don't know what the number is, but I keep hearing that, when people do this, that there's only a certain subset, right? And obviously, the older the cells are, if you have an older population, I believe it's harder and harder to reprogram them, and you get only a smaller amount that you can reprogram from that population, which suggests that there is, that there's too much information loss could happen. You can't get reprogramming to happen. When it comes to generating, viable offspring or babies, we know that, not all pregnancies are successful. At a certain stage, I believe, at a certain point of, after conception, you have a very high rate of spontaneous, abortions or miscarriages. It's up to like 50%. And most people don't even know it happened because it happened so early. And of course, men generate millions of sperm and only one actually is, impregnates fertilizes the egg and you get a zygote. As the eggs themselves develop, right, you basically have not all of the haploid eggs essentially become are the ones that will get fertilized. The other ones become polar bodies, and something happens to them. They don't actually end up. So the point I'm making here is there's a certain winnowing process where I think if certain cells are too far gone with information loss, they just don't make the cut, right? I think that probably applies to the developmental process. It applies to somatic cell reprogramming. So the point I'm making is that it's not a closed system, as Steve mentioned. That entropy is, or with hydra shedding cells, that entropy is getting removed out into the environment to maintain, a small select lucky group that is able to re-maintain and reestablish that information. But we can't ignore the fact that that entropy is being jettisoned out there, right? So there's no free lunch here. It's not like a perpetual motion engine here. It's an open system, and the system has developed a variety of ways to keep pushing that entropy out, so it can basically maintain a subpopulation that can keep going in perpetuity, right? So that's my take on this.
Oh yeah, I'm going to circle back on what you said earlier about how potential gains of lifespan. I know you were just spitballing, obviously. Don't quote you on exact numbers. I mentioned in the chat something that Irina and I were speaking about, where you reprogram the stem cell niche with young blood, or blood that's had the negative things removed. It's interesting. How that works as well, because there's epigenetic programming is occurring there, and I just think it's interesting. So something Irina said when we were talking was that there's reason to think that such a reverted state in the blood would actually persist for some time, because humans, not mice, have a lot of they've got a lot of feedback loops and a much more robust repair system. So to add to the mix of speculation here, whilst we might not see such huge gains in lifespan in more simple organisms like mice, we might see significantly larger ones in humans with all the feedback loops and systems that we've got. So, could it be, as Irina suggested, about when you reset the blood, the state persists? When you reset the epigenetic state of cells, en masse taken as a whole, does that sort of persist longer in humans because we're more robust? Let's face it, we accumulated that much epigenetic damage over a very long time. I mean, it didn't just happen overnight, right? So if we reset all the systems so they're nominal again, would it take a similar amount of time to accrue that amount of damage again? I would probably suggest maybe.
Yeah, I mean,
there you go. That's absolutely vague as possible.
Well, that exact question was raised in a review paper I read recently on partial reprogramming, and that's an unanswered question right now. Is there diminishing returns with reprogramming? I think
probably not.
I say probably not if we're just looking at reprogramming and we're just looking at the cells that are being reprogrammed that have enough information content left that you can keep reestablishing it. For the organism as a whole, I think there are diminishing returns, because I think there will be other forms of entropy accumulation that epigenetic reprogramming is not dealing with, which will eventually come back and through a negative feedback loop, start repressing those gains.
All the more reason to bank your DNA, right? So then you've got a master template for which to create more stem cells to then transplant stem cells back to your body and start the cycle all over again. So there's going to obviously be loss of stem cells. I mean, stem cell depletion is one of the proposed reasons we age. As you say, eventually you're probably going to run out of stock, but I reckon you get a few more miles on it. So, I'm quite bullish on reprogramming. And to just to circle back to what we talked about ages ago, one of the traditional limitations of OSKM, or even 3 factors, I think they've even tried 2, was the actual viral vectors are not particularly effective, not to mention being very, very prohibitively expensive to do that form of traditional gene therapy. But it looks like people are more and more sort of moving to the mRNA, like the vaccine delivery style, which is really good, because we know from COVID that it's infinitely easier to build that at scale, and it's quite accurate. So people say, "Oh yeah, it'll only be for the rich." Yeah, just like just like the COVID vaccine was only for the rich, right? So I honestly think we're at quite an exciting time. I'm seeing I'm actually seeing senescent cell therapies using mRNAs as well, and one that resets Sensica with Lorna. You probably remember Lorna spoke at one of our conferences. I think it was a year or 2 ago. Well, they're resetting. What is it they're doing? They're resetting something that causes the DNA to unravel, off the top of my head, which is interesting. And then I believe they're using an mRNA delivery system. So, and they're much cheaper and easier to build at scale. So I really think we're going in the right direction with this. It's getting pretty exciting. Because viral vectors, let's face it, they're not great. They've got a lot of limitations. So, and you mentioned that not all cells are reprogrammed. You're probably right about the fact that some of it maybe has lost too much information, but we just don't know, because they're even finding ways now to make reprogramming more comprehensive, and they've combined it with senolytics and found that boosts stem cells as well. So yeah, there's a lot of stuff that's interconnected here. I think we're not that far away from actually working some pretty big stuff out. Call me an optimist if you like, but that's what I feel.
I mean, at the end of all of this, I'm not saying now or probably in our lifetimes, we're just going to have to, once we have enough information, genetically modify ourselves at at the germline stage, that's the only way that we'll have humanity live substantially longer. When I say substantially, I mean essentially thousands of years or more. So this is now going way ahead. So good question.
That's
there.
A lot of tax returns.
Well, good question there. We can have philosophical discussions now. Is like, are we essentially human at that point, right? Are we
Yeah.
We can say, are we if you have a subset of humans now that
I'm going to use the ship of Theseus argument, Oliver, to count that one immediately and say, or like the old analogy of, we've had a broom, it's been in our family for 300 years and it's been passed down. We've replaced the handle, we've replaced the bristles, and we've replaced the string many times over that 300 years, but it's still the same broom. And I think that's actually true of what you've just said. Are we still human? Yeah, I would say definitely. A case in point I would give you is, when I was 20, I was a very different person to when I am, to the age I am now. Was I the same person? Yes, broadly speaking, but we've replaced a lot of my bristles since then. Over to you.
Yeah. Well, I was just more referring to the fact that if you genetically modify an organism to have to have a greatly extended lifespan by really putting in novel systems, then that in of itself would be corrupted if you procreate with humans that don't have such an optimized system, right? So then you're branching off. It's a speciation is taking place at that point.
I feel like a conversation about Gattaca is coming here.
No, not Gattaca. I'm talking about more kind of fundamental evolutionary speciation. self-directed evolutionary speciation. Not so much. Yeah, I guess some Gattaca components there, but
It is a little bit like you had the and I haven't seen it for years, but if I recall, the people who were unmodified were called the invalids.
I guess. Yeah.
I think they were the invalids. Um, eh, and basically treated very much like second-class citizens. I think it's a great basis for a science fiction film or a book. But no, are you
I'm
still human? Yeah, I'm going to say yeah. I mean, and look, Oliver, being a human's overrated anyway, mate. I mean, I haven't been a human for years, so I'm told.
Alrighty then. Okay, so
It's
now,
about that time in the afternoon where we start making the jokes.
yeah.
But no,
Um,
I mean, very interesting.
so
As you
yeah,
say,
we need to quantify. I think the entropy loss and exactly, at least focusing right back again, circling back to the epigenome. I'm really interested in finding out uh, how that information is still maintained to allow reprogramming to happen, and how it's still encoded. And how that affects the efficiency of reprogramming, because there's a lot of stuff published right now that's still kind of, a lot of painstaking experiments, but there's still, when people talk about the efficiency of reprogramming and how reprogramming exactly happens and how much information is actually lost, pretty much every single paper, it's still qualitative hand waving, right? It's sort of, there's nothing, there's no real concrete numbers being put here, where we can actually, get a percentage, and quantify the loss of information or the extent of good information that's still required, and where that information is encoded. And I think, I think the next series of experiments should address those issues.
yeah, and also what they're doing right now is obviously working on more complete reprogramming, partial reprogramming, better target delivery, which is then obviously going to make it easier to do, less treatments, again reduce the cost. mRNAs seem, and I keep going on about them, but I honestly think they're a massive step forward. And as the pandemic recently showed, they are very, very scalable and very, very easy to deploy compared to other approaches. So I think that, as you say, that the studies need to be done, the delivery needs to be improved, and also the completeness of the reprogramming, that also needs to be improved. And I think that's really, that's the big focus right now. Is we know in potential that it could work because we've done it a number of times, but we've got to do it efficiently. And I think that's what people are doubling down on now. So I think we've got a long way to go. Personally, I'm writing an article at the moment, which will be out in the next few weeks, which is we're going to take a look at the technologies that are sort of near term, mid term, and long term. And spoiler alert, guys, I am going to say that I think, I mean, I wish it wasn't so, but I'm going to say I think the partial reprogramming is a much more further out prospect. I honestly think you're looking at 20 years. But I think we'll get there.
Mm-hmm.
I mean, if nature can do it, so can we.
Exactly. Anyway, great topic. So yeah, it's good to should do this a little bit more often, which is basically try to cast a wide net and try to focus a little bit more on the theoretical aspects, because we certainly focus a lot on the, the meat and potatoes, so to speak, of primary literature, and really drilling down into specific experiments. But then you kind of, lose the forest for the trees. So it's good to kind of zoom out, get that 30,000, foot viewpoint, and not get too lost, and try to keep in mind kind of the general themes that I see kind of congealing, right, as this field gets on a firmer footing as the years go by. Hopefully sooner than later, because we're all outrunning entropy, and eventually it's going to catch up. So
Nice.
yeah.
Unless we got, as David calls it, we out regenerate it or out rejuvenate it, and in his book he calls it the Wolverine scenario, which I quite like actually. I know it's been proposed in the past before this, the idea that regeneration or rejuvenation can occur faster than the organism damages itself and ages. So it's a possibility. I mean, you can sign me up for some Wolverine stuff. Not necessarily the claws. I don't think that's necessary, although it could be useful in the garden.
I kind of like the analogy with Lewis Carroll and the Red Queen, which is the statement that she said in the book, which is supposed something to the effect that you have to run faster and faster just to stay in the same spot, right? Which is basically what cells are doing. Just have to keep running faster, stay in the same position, the same spot. It's not easy to stay. It's not easy to stay in the same spot if you think about it.
That's true. So yeah, interesting stuff though. Would I say I was totally convinced? I think that theory has its place. In the grand scheme of things, eventually, Oliver, we will get there. We'll have that grand theory of aging, or that general theory of aging, I should say. It's not fair. Physics have got all their general theories, so it's about time we've got our own. These darn physicists swanning around like celebrities. It's unacceptable. Actually, it's quite a lot of physicists who are involved in airfield now as well, like people like Andrew Steele. Who was the other one?
Well, if it wasn't for physicists, we'd never have molecular biology. So,
Yeah, although I do
think,
want.
yeah.
I like what a physicist once said. They said, with physics, it's easy. The more you drill down, the simpler it gets. With biology, it's the opposite way around. The more you go into the weeds, the more complicated it gets.
Yeah.
It's probably true.
Anyway, with that, I think I've exhausted this topic at this moment. I will think about what we've just said, and I will, dive back into the literature searching and see if some of the questions that I had are at least partially answered by some publications that I've, not noticed in my earlier searches.
Yeah, I mean, and we could look at delivery and or the completeness of partial cellular reprogramming. We've got the experiments of people like Jean-Marc Lemaitre, the original Yamanaka stuff, and we can potentially do a whistle stop tour and see how things are coming along and get to the end. And I'm not going to mention mRNAs too much, but yeah, we could talk about that too.
Okay, and we'll talk about it maybe at our next journal club, which is, I'm not sure when, because we had to postpone. Are we having it at the end of February? Or are we having it at the end of March?
Well, we usually try and aim for the end of the month, but sometimes we have unforeseen circumstances. But do bear with us, and we will get to it. I would say probably in a few weeks' time. Let's have a look. Probably the 23rd. So 3 weeks' time, I would say, if we want to get back into the pattern.
Okay, so so I think maybe we'll swing back and do a primary paper. So whether it's going to involve partial reprogramming, I don't know yet. So stay tuned. All right.
Yep.
So
Right.
that's all I got. Thanks, everybody.